Hey y'all, welcome back to another episode on genetics. Today what we're going over is a part of chapter 4, which goes over variable phenotypes. And the reason why this part of the chapter is really important is because not every single gene in existence has those three characteristics that we talked about yesterday, which was complete dominancy, incomplete dominancy, and codominancy. So in a perfect world, there would be only three, but in reality, there's multiple different kinds of interactions that can occur between different genes. And then between these interactions, you can also have some differences in, the allele numbers, which affects the allelic series. You can have some alleles that basically, when you're homozygous dominant for them, you would die, and that will also skew the data that you collect for a particular trait. So that's another important part, and there's other examples that we're going to go over here as well. But the most important one for now is allelic series. So if you remember back from the beginning, that every gene in this world has almost an infinite number of alleles that you can have possible for that gene. And because of this, in nature, you always have 6, 7, 8 different alleles that contribute to a different phenotype. And these different alleles, they interact with one another in a different way. So it could be that, let's say we're looking at gene A, and it doesn't matter what gene A is encoding for, but we could say that we have a big A. We can say that big A is dominant to little a. But then let's say we have another allele. We're going to call this one big A plus. So let's say little a is incompletely dominant to big A plus, but both of them are recessive to big A. In another part, we can also say that these two are also dominant to another allele. We can call that big A1. And something like that. So that's basically the point of an allelic series, is to introduce you that there's almost an infinite number of alleles possible for a particular trait. And because of this, they all interact together in a certain way, where an individual that is diploid could be big A plus, and that'll have a different phenotype if you were little a big A plus. Something like that. So you can have a completely different phenotype based on how these genes or these alleles interact with one another. And that's basically the whole point of allelic series. Lethal alleles, on the other hand, is basically, like the name suggests, if you have this lethality allele, you're basically going to die. But that's only the case if you're homozygous dominant. So basically, if you have a situation where you're crossing big A little a with big A little a, 1/4 of your offspring are little a, and then this 1/4 will be basically dead. So basically, that's all lethal alleles are. It's pretty simple, easy to spot too. And then now this is where we get into the more complicated stuff, where we talk about sex-limited traits and sex-influenced traits, and then pleiotropy as well. So let's talk about sex-limited traits first. So sex-limited traits are basically traits that are... So basically these are traits that are essentially turned on when a specific sex inherits the allele. So basically what this means is that, let's say we have let's say we have all of these individuals, like both males and females. These traits are only involved in autosomal genes, such as big A or in little a. So let's say that in our example that a female inherits, say, big A, little a genotype, and a male also inhibits the same genotype as well. But since the male has that little a allele, and it also has testosterone that the male produces, that testosterone is going to interact with the little a allele, producing some sort of a different phenotype compared to the female individual here. So what you can write this down is that the testosterone and then we can say that little a also interacts in this pathway too to generate some sort of a phenotype. And then as long as the male always has that little a phenotype, they will always have that particular phenotype that we're looking for. But the female, on the other end, she doesn't produce any testosterone. So because of this, the pathway on the right will not be able to be accomplished, and you wouldn't get that certain phenotype at all from the females. And keep in mind, this has nothing to do with sex linkage, because sex linkage is almost always correlated to a gene that is found on the X or the Y chromosome. These traits these genes are always found in your autosomal chromosomes. Okay? Cool. So the next one is sex influence traits. So it's basically the same thing, but a little bit different, where the sex only has a slight influence on, not a slight influence, but the sex has the influence on your trait. It isn't just if you're male or female, you're going to be a certain phenotype. It's essentially like the figure on the right over here, where if you have a male, which is right here, if you have a male that has the B2 allele, like we said with this example, little a over here, you're going to be bearded. And same over here as well. But the female isn't going to be bearded at all. But this changes when both the female and the male contain a B2 genotype. Then because of this, that female now has that bearded phenotype. So essentially, the way how you can describe this is that, like gene dosage and how we talked about a certain gene produces a certain amount of enzyme and stuff like that. If an individual that is female, she doesn't have any testosterone, so she needs another replacement of little a in order for her to have that same phenotype that the males have. So that's basically the general gist of a sex influence trait, is that the sex only influences a phenotype, but not completely though. It doesn't matter if the female female inherits the allele. She just needs two alleles for it to have a certain phenotype as the males. So it isn't only limited to a particular sex. It can be expanded on. And then pleiotropy, this one, a lot of people make it harder than it has to be honest. Pleiotropy is basically one gene affects two traits. That is all pleiotropy is. It's basically just a single gene affecting two different traits. And one of the things that you're going to see with pleiotropy is that, let's say we have two different traits which are big and tall. They're always going to be big A. And then if you're small and short, you're going to be little a. So in pleiotropy, if you're crossing big A, little a with big A, little a, what's happening here is that you're getting a 3 to 4 ratio, a 3 to 1 ratio actually. My apologies. Big A whatever, and this one will be little a. So what's happening here is that all these individuals over here are big and tall, and all these individuals are small and short. So if you have a data set where you're trying to look for cases of pleiotropy, one of the biggest indicators for pleiotropy is that you're never getting a blend of different traits together, right? We're never seeing a case where we're getting a individual that is big but also short. We're never getting that. Oops. We're never getting that combination, but we're also never getting a combination that is small and tall. So we're never getting that either. So that'll be your prime indicator that something is pleiotropy. So let's go over some examples that you might see on an exam. And we've already talked about this problem before during the sex linkage question in chapter 3. But essentially right here we have males and females. They're basically pretty much equal, and they have the exact same phenotypes as well, right? So you have yeah, so basically both males and females are equal in phenotypes, right? These two are basically equal, and then these two are also basically the same thing. So basically what we need to do is figure out the mode of inheritance. And just looking at this, we can combine these first to actually make it a little bit more simpler, because there's a lot going on. So if you can look at this following data set, basically what I've just done is I've combined these two together and these two together in a single phenotype. They're expressing the same numbers of males and females alike. But now what we need to do is we need to figure out, looking at this particular phenotype, what kind of mode of inheritance is this? And by looking at it, we can tell that it's basically a 3 to 1 ratio. And because it's a 3 to 1 ratio, and looking at the phenotypes that we're never getting a blend of phenotypes that is long hair and small, but we're also never getting a blend of phenotypes that is short haired and tall as well. So because of this, because we're never getting a different blend between these, this is also the case of pleiotropy for this particular trait. So this is pleiotropy. So hopefully that made sense. And then the next part, this is where a lot of people get confused on. I've seen this question on a couple of exams, and this is a lot of people, they get confused on because there's a lot going on, okay? So what you have here, you have a gene called Amarillo, and you have several different alleles, so we're looking at an allelic series. And the key to these kinds of questions is you always want to write down what your allelic series is and how they interact with one another to give you a certain phenotype. So over here, it tells you that big A, which has a phenotype of Lamar, will be completely dominant to every other allele. So we're going to write down that big A is completely dominant to whatever, and we're going to write down that this is the Amara phenotype. Next, it tells you that big A plus is Omar and is incompletely dominant to A minus, which is Lamar. So we could say that A plus is basically incomplete dominant to A minus. That's incomplete dominancy. So when you're big A plus, you're the Omar phenotype. And then when you're the big A minus, you are the Ramar phenotype. And then it tells you that little a palmar is recessive to every other allele. So we're going to write down that these are completely dominant to little a, which is Ramar. Boom. So now you have sort of a key that you can use to figure these questions out. And we've basically done the hard part. Now we need to look at the problems that we have. It tells you that you have a completely unknown set of Amarillos, and you're trying to get the following proportions of genotypes. You're getting 28 Amars, so you're getting 14 Ramars and 13 Omars. So essentially what you're getting here is a 1 to 2 to 1 ratio. So basically 1/4 of your individuals are big A, whatever, right, for the Amar. 2/4, or excuse me, that was a 2/4, my bad. 1/4 of your individuals are Ramars, which are little a, correct? And then you have another 1/4, which is Omars, which are big A plus, little a. The reason why it cannot be big A minus, it could also be big A plus as well over here too. So we can just write down, big A plus, or big A plus, little a. But the reason why it's not big A plus, big A minus is because if that was the case, then we'll get a completely different phenotype, which would be this would be Palmar, my bad, y'all. And then this one, yeah, so if you're big A minus, you're going to get a phenotype of Ramar. And then A minus is Lamar right here. So yes, so basically being big A plus, big A minus is Ramar. So yeah, so what I did here, I made a mistake. This one should be big A plus, big A minus, because I didn't read the question, obviously. I didn't do a good job at reading it. And then you ended up with 13 Omars as well. Okay? And the Omars can be in this arrangement. So now what you need to do is you need to figure out what kinds of genotypes were combined together to form this data set that you have over here. So first of all, what you should do is you should always begin with the beginning, which is big A. Start with the one that's the most complete dominant. So we're going to write down that parent number 1 has big A. And this will be parent number 2. And then from here, what you want to do is you want to go backwards and try to determine what kinds of genotypes are you getting and in what order. So for example, over here, what you're getting is essentially you're getting an individual that is big A plus and big A minus, right? That big A plus, this one right here, had to have come from a parent. And then this big A minus had to have come from a different parent as well, right? Because you can't get a genotype without getting the chromosomes, getting the gametes from a parent. So therefore, these you can consider as 2 different gametes from 2 different parents, and that will guide you towards what the correct genotype is for both of the parents, okay? So hopefully that kind of makes sense. So yes. So we're going to say that parent number 1 over here has the big A plus allele. And then the parent number 2 has the big A minus allele. right? So this had to have come from mom, and then this one had to have come from dad, right? And then now it tells you that you have a situation as something like this, where we're getting a 1/4 probability of getting an individual that is an Omar, which is basically big A plus, or you could be big A plus little a. So what we need to do now is we need to figure out, it's basically just guess and check at this point. What you need to do is you need to figure out what the genotype of parent number 2 is for you to satisfy this following cross. So what we can do is guess and check. We're going to say that this parent number 2 has a little a allele. So if we cross these together, essentially what we're getting here is a 1/4 chance to be big A from this gamete, and then this gamete right here, big A minus, right? And then 1/4 chance to have big A, little a, 1/4 chance to have big A plus, big A minus, and then you have a 1/4 chance to have big A plus, little a. And then if you have certain phenotypes that are the same, such as these two over here, they're the same phenotype because they have both big A's and they're all completely dominant. That will give you a ratio that you have these two, you'll combine using the sum rule. That would be a 2/4 ratio for big A whatever. And then you would have a 1:2:1 ratio. So these were the original genotypes of the original parents. So hopefully that kind of made sense. Always apply this following rule over here, where you look at the product that you have, which is big A plus and big A minus. Those two had to have come from different parents, and because they came from different parents, you now have a clue on how to align your genotypes for each particular parent. Okay, and then as a bonus problem, you have a Lamar, which is, let's see, big A minus, big A minus, we can say big A minus, little a. I think that one is not fully well written out. And then you have an Omar, which I believe is big A plus, big A plus. Oh yeah, you could also be big A minus. And then the question is asking you for the proportion of this particular genotype, little a, little a. So we're going to say in this situation, we're going to say big A minus crossed with that big A plus, little a. So then what you're going to end up here is 1/4 of your offspring will be little a, little a. So hopefully that all made sense. Thank you for watching. Hope you all have a good day.